Organic compounds. We've already mentioned how organic compounds will contain carbon in them. So now we need to kind of expand on what these organic compounds are. For our bodies, we need to go through 4 main macromolecules, looking at carbohydrates, lipids, proteins, and nucleic acids. This video will focus on the carbohydrates and the lipids primarily. Next video will deal with the proteins and nucleic acids. So carbohydrates. Now, while there's not just one single carbohydrate, there's many different types of carbohydrates. The one distinguishing feature, all carbohydrates have a 1:2:1 ratio. They all have 1 carbon to 2 hydrogens to 1 oxygen. So 1 carbon, 2 hydrogens, 1 oxygen. This 1:2:1 ratio is basically the main foundation of all carbohydrates. And most carbohydrates in our bodies are used as an energy source. They'll be used to make energy for our cells, for our organs, basically for our bodies. Now, it's not the only purpose, but carbohydrates really, a lot of them are used for energy purposes. So the carbohydrates, the category called monosaccharides. Mono means one, so monosaccharides are one single ring. If you notice, we have glucose, fructose, and galactose here. All have a ring-shaped structure. We have deoxyribose. Deoxyribose is the sugar of DNA. Ribose, the sugar of RNA. But you'll notice they all have one single ring-shaped structure. Now, sure, some have 5 points, others have 6 points in the ring. The idea there is it's a 5-carbon ring or a 6-carbon ring. But they still have the same basic idea. If you're trying to count the carbons, things where there's corners like here and here, those would be symbolic of carbon based on that nomenclature there. So if there is just an angle, then that angle is where you're looking at being a carbon. So these are all monosaccharides. If you put them together, 2 monosaccharides make a disaccharide. Now these disaccharides, like sucrose, lactose, and then maltose, they are 2 monosaccharides bound together to make a disaccharide. If you remember back to previous videos, the idea of dehydration synthesis. You can take the 2 monosaccharides, they go through dehydration synthesis, and they make a disaccharide. For example, this lactose you see. Well, lactose might be familiar as the milk sugar that's found in dairy. So we have our disaccharides. And then we have polysaccharides. Polysaccharides, poly meaning many, are going to be 3 or more of these rings held together. Some examples you see here from amylose and amylopectin. Those are going to be some starches. We have our glycogen. Glycogen, that is going to be our body's storage unit of how we store glucose. Each one of these individual rings, one there, each one of these rings is a glucose molecule. So you can see how we're storing numerous glucose molecules, all in this one molecule of glycogen. It's our body's way of storing glucose for future use. And then cellulose, last one, that kind of interlocking woven set for fiber. So these different polysaccharides, some our bodies can break down and utilize, others our body can't really utilize for an energy source, but they still are important in other functionalities. All right, let's switch over to lipids. Now, lipids as a category will still have carbon, still have hydrogen, still have oxygen. The difference is now, it's not the same ratio. It's not the 1:2:1. The 1:2:1 ratio is only in carbohydrates. In the different lipids, you don't have a ratio because they different depending on which one you're in. See, there's 3 types here. Triglycerides, or known as neutral fats. Moving from triglycerides, you can move down to the next one, which is your phospholipids. And from phospholipids, you go down to the bottom one, which is your sterols. Each one of these three types or categories is found within the overall category of lipids. Let's start with the triglycerides. Now this image is showing the triglyceride here is made up of glycerol and 3 fatty acids. But if you notice on the one, you're looking at the H's and the O's are all in blue. That's because before the triglyceride is built, you have to remove those water molecules. That means you go through 3 different reactions of dehydration synthesis. Ultimately, that gives us the glycerol backbone and the 3 fatty acids coming off of that glycerol. That is your triglyceride, tri-3-glyceride. So 3 fatty acids, 1 glycerol. That is your triglyceride. But these fatty acid chains, if you notice it's Ch02, Ch02, Ch02. Remember, carbon has 4 of the electrons in its valence shell, which means it can form 4 different bonds. In this structure, we'll notice that a carbon binds to a carbon, so you form that carbon-carbon bond, then another carbon-carbon bond. But that still leaves 2 more bonds to be made. We make a hydrogen bond and a hydrogen bond off there. So we end up with bonding 2 hydrogens and 2 carbons for each carbon molecule. Now, when I said hydrogen bond, that wasn't like a hydrogen bond we talked about in bonding types. This is carbon bonding to hydrogen, for example. You'll notice that there is a saturated and an unsaturated. The saturated means every carbon has its maximum number of bonds possible, while the unsaturated is going to say, we're not going to have our maximum amount of bonds. Right here in the red, you're going to notice there's a double bond. That's a carbon forming a double bond with another carbon. It's not having its maximum amount of attachments or bonds. If it was only a single bond, there could have been 2 more hydrogens bound there. But because it was a double bond, it is referred to as unsaturated. Unsaturated because you don't have the maximum possible amount of hydrogen bound. So sometimes we'll hear about saturated or unsaturated fats. This is what they're referring to. Saturated means maximum amount of hydrogens. Unsaturated means you can actually bind more hydrogens. Phospholipids. Now, phospholipids is the second category. In phospholipids, we still have the glycerol backbone. We still have 2 fatty acids. But instead of the 3rd fatty acid, now we have this phosphate group over here. So we have replaced one of the fatty acid chains with a phosphate group. Why that's important is these phospholipids are what we found in our cell membranes. You create this double or bilayer of phospholipids. And by adding in this phosphate group on one side, it allows this molecule, this phospholipid, to be hydrophobic on one side, hydrophilic on the other side. So these actual tails down here, these 2 different fatty acids, in that case, they're more of a hydrophobic. They don't like water. But the phosphate group, which is the head group, that is now hydrophilic. It can interact with water, no problem. So this molecule is very important in our bodies because it's what makes our cell membranes. The third category is steroids. Now, steroids are a little different because they still have the ring shape, just like our carbohydrates. But notice the rings are interlocking. There's no space between them. So our steroids usually have the interlocking rings without that space in between. Steroids, you'll think things like cholesterol. You'll have an idea of that. So there's different types of them, but we'll hit on those a little more later. That's the third type of the lipids. Prostaglandins. Now, prostaglandins are kind of a separate category on their own, but these are signaling molecules. You'll notice it's more of a complex molecule shown here, but I just want to put that term out, prostaglandins, so it's aware when the future comes back, yeah, I've heard about those before, it's part of this, so you can kind of place them where they go. So prostaglandins, more signaling molecules. So lipids, carbohydrates, 2 of the 4 main macromolecules that are organic compounds.